Welcome to Pharmacognosy and Metabolic Engineering NPTEL online certification course. So, today is lecture 14. So, where we will cover engineering tropane alkaloid pathway in plants, further advancement, and then engineering tropane alkaloid pathway in yeast. So, let us go to the next slide. So, first we will start with the complete enzymatic route to tropane alkaloid biosynthesis. Then we will come to individual case studies like enhancement of tropianone and litorin in engineered root cultures. Next, we will talk about engineering tropane alkaloids by modulating ornithine decarboxylase activity, and once we finish that, I will go back to the first plant metabolic engineering paper which published in 1990, and then we will talk about tropane alkaloids in yeast. This is one of the most exciting area. So, here I will discuss the advancements published in 2020. So, start with, so in the previous class I have spoken about the enzymatic route to tropane alkaloid biosynthesis. So, what is showing here is almost all the steps of tropane alkaloid has been elucidated. So, this is a paper, outstanding paper published in New Phytology Journal in the year of 2020, where they have characterized these two enzymes. One is this UGT1, that is glucosyl transferase, which act glucose to the phenyl lactate and makes phenyl lactyl glucose, and the second one is the litorin synthase, which catalyzes the reaction by joining phenyl lactyl glucose with the tropinone and makes litorin. So, this particular work or this particular publication basically describe these two steps, and along with that more or less the enzymatic route to tropane alkaloid biosynthesis is now available. So, the next interesting case study which was published in Industrial Crops and Products in 2022. So, where enhancement of tropianone and litorin biosynthesis in root cultures of Atropa belladonna. So, in one of the previous classes I have spoken about the root cultures, co-transform root cultures. So, that means these are hairy roots. So, these hairy roots contain the genes for tropianone and litorin biosynthesis, which was inserted using a binary vector approach, and as a result of that the hairy roots are expressing these genes, and this is the hairy root of Atropa belladonna. That means, the tropane alkaloid pathway is operating. So, there may be additional copies of genes, these genes can be inserted, and what happen as a result of that. So, let me go to the next slide and then we will back to again. So, there are 4 genes they have considered here. In the first case they have taken this polyketide synthase, which converts N-methyl pyrolinum to this intermediate MPO, and the second one is the cytochrome P450, which converts the MPO to tropinone. So, in one set of experiment they have co-transformed these two genes together, and the other set of experiments they have taken UGT and LS, and in the third set they have added all these 4 together. So, 3 approaches were taken. Let us go back to the previous slide. So, co-overexpression of PKYS, that is the polyketide synthase and cytochrome P450 genes, led to increased tropinone and tropine content, and which is basically obvious, because when these two are overexpressed, then as a result of that there will be more tropinone and of course tropine. Co-overexpression of UGT and LS led to increase in hyoscyamine and scopolamine content. So, that means, when 3 and 4 genes were overexpressed, in the co-transform hairy roots, that leads to more lithodin formation, and as a result of more lithodin the pathway push towards the end product, and as a result they see the product formation there. And upon 4 genes co-express, the highest level of tropane alkaloids are obtained. So, that is also obvious, and what they said that the tropinone and the lithodin biosynthesis were identified as 2 rate limiting steps. So, once you can have these 2 intermediates produced in higher amounts, then the final product will be enhanced. Another interesting work which published in ACS, American Chemical Society Synthetic Biology, in the year of 2020. What they have done? They have taken more or less a traditional approach. So, there they try to modulate the pathway by expressing ornithine decarboxylase. So, ornithine decarboxylase is one of the early enzymes of the pathway which converts ornithine to putrescine, and then subsequently there are so many steps as I described before, and that leads to the formation of ornithine to putrescine, that step, and then that led to the formation of the tropane alkaloid. So, let us see their approach. So, what they have done, they have established Atropa belladonna root lines. One is that they have suppressed the ODC by RNAi approach, so that ODC gene will not function, and as a result in the root cultures the suppression greatly reduced the production of putrescine, N-methyl putrescine and tropane alkaloids. Let me see if I were drawing here. Okay. So, when the ODC is suppressed, as a result of suppression, the final products will get in reduced amount, will accumulate in reduced amounts. And they have also used the whereas when they overexpress this, then that led to significant increase in the biosynthesis of putrescine, N-methyl putrescine and hyoscyamin. So, this is a straightforward interpretation, that means, if the gene expressed and if the gene expressed under the control of a strong promoter, so it produces more mRNA, that means more protein, when more protein is available. So, if substrates are available, so it will convert the substrate to products, and as a result of that what we see, that ultimate end products are produced. So, this is not always the case, because when one try to over express the early genes of the pathway, the result sometimes may not reflected in the end product, but in this case they were successful, they found positive results with the end product accumulation. Similarly, they also attempted to make the transgenic plants, and the transgenic plants also showed the similar results, that is higher production of hyoscyamin and anisodamine. So, anisodamine is nothing but the hydroxyhyoscyamin. So, that is renamed as anisodamine. So, that is produced in transgenic plants of Atropa belladonna expressing ODC. So, from this now I go back to the first report in the world, basically the first metabolic engineering paper published in the world. So, here what they have done, so this is a group from Norwich, UK. So, Hamill, they published this paper in the reputed journal Plant Molecular Biology. So, what they have done, in those days they were involved in the characterization of the early steps of the tropane alkaloid biosynthesis. So, they were using different plant systems, Dhatura, Nicotiana, Hyoscyamus. Okay. So, what here they have attempted, they have taken yeast ornithine decarboxylase. Now, this source was from yeast. So, they had in their mind that if they take a heterologous gene, then there are chances of gene silencing will be less than to take the plant gene itself, and the yeast ornithine decarboxylase is the same function which converts ornithine to putrescine, and they express it in the tobacco, and as a result of that they wanted to see what happens to the nicotine content. So, nicotine is the end product, and you see that nicotine requires nicotinic acid addition along with the N methyl pyrrolinium ion. The pathway is short. So, therefore, ODC overexpression, it is possible that as a result of ODC overexpression you may see a positive impact on the nicotine content. Okay. But it was not that easy unless they add some enhancer sequence upstream of the promoter, it did not work properly. So, when enhancer sequence were added upstream of the promoter, so enhancer sequence plays important role. Basically, it communicates with the promoter through some proteins, maybe transcription factors. So, that allows the promoter to function more efficiently. As a result of that, transcription will happen more effectively, and you see the enzyme protein formation. So, when they have used this enhancer sequence, they found, and what they found, that these cultures were capable to accumulate nicotine. That is a positive result, but the frustrating point at that time was that even they used a very powerful promoter, they have used an enhancer sequence, even then the increase one is only of the order of twofold. So, what they thought that there will be a havoc increase on the content of nicotine, that did not happen. So, which basically suggests that the flux control is somewhere there in the downstream of the pathway. So, just putting upstream enzyme, overexpressing an upstream enzyme may not be able to do the job, but nevertheless it demonstrated for the first time that it is possible to manipulate the specialized pathway by overexpressing heterologous gene. So, that means genetic manipulation for plant specialized metabolic pathway is now become reality after publishing this paper. What we see much later in 2003, another paper published by Hamill. This time Hamill was not in Norwich, so he moved to Australia, Monash University. So, here what they did actually, they downregulate the PMT activity. So, PMT converts putrescine to N-methyl putrescine in the tobacco plant. So, in the tobacco plant they use antisense approach. With antisense expression of PMT, what will happen, that it will disrupt the function of PMT. So, there will be less formation of N-methyl putrescine, and if it is so, then what is going to happen, that the nicotine production will get affected, because it is tobacco. Remember, tobacco is producing the nicotine alkaloids, but what they found to their surprise, that of course, the nicotine production is getting down regulated, but that in turn uplifted the formation of anatamine. So, anatamine is another tobacco group of alkaloids. So, what happens that when nicotine formation is down regulated, that means the nicotinic acid, which is one of the substrate for the production of nicotine by joining with N-methyl pyrrolinium. So, when this pathway is ineffective, so that means there are lot of nicotinic acid, the nicotinic acid which is produced for the production of nicotine is remain unutilized. So, in other words, if there is one enzymatic route exists here, which joins nicotine with 1-methyl. So, that enzyme activity will get shut down, maybe as a result of feedback inhibition, whatever. But what happens, this leads to more nicotinic acid accumulation. When nicotinic acid is accumulating more in the cell upon antisense suppression of PMT. So, this nicotinic acid is now converted towards the anatamine. Along with that, also some anabasin is formed. So, elevated anatamine levels in antisense as a result of consequence of relatively oversupply of nicotinic acid in the absence of adequate level of N-methyl pyrrolinium ion. So, and this is also a very interesting paper, although and interesting from this point, that normally in the tobacco, wild type tobacco, you hardly see any anatamine formation. It is all dominated by nicotine. Okay. So, this is the first time that by manipulating the pathway it is possible to make accumulation of these alkaloids in the tobacco in significant amount. Now, let us try to see the things in a different way. So, there is a need for production of engineered tropane alkaloid in alternative organisms. Why? Because the tropane alkaloids are harnessed from the plant to fulfill the demand of pharmaceutical industries. Okay. And one cannot depend on chemical synthesis, because it is a very complex stereochemistry involved in it. So, therefore, intensive cultivation is required, and that is basically the backbone for supplying the alkaloids to the pharmaceutical industry. Now, point is this agriculture based supply sometimes poses risk to the public health. Why? Because sometimes overall increasing demand of tropane alkaloid based medicines already resulted in recurring supply shortages. Sometimes demands are so high it cannot be fulfilled by the supply of the wild plants from the nature, that is a problem. And second, that sometimes natural calamities, wildfire, war can threaten the global supply. And third one, global crisis like what happened a few years back, this COVID-19 pandemic, that can also threaten local ability owing to this, and as a result of demand spikes and disruption of supply chain. So, that means, if one needs entirely one is entirely dependent on the supply of the plant from the wild sources, then these possible problems one may face. Okay. So, the what is the alternative, what is the way out? The way out is this: if such tropane alkaloids can be produced in an alternative system, which is basically free from any regional local demands, free from geopolitical dependencies and robust environmental socio-economic turmoil, then that would be great. So, how then what we can think of? We can think of microorganisms. If the pathways can be transferred in microorganism, then the microbes can grow anywhere in the world, and you need not have to depend from the wild to get the supply of it. But this is a great challenging task, because as we see that the pathway is very complex. This is a multiple step pathway and it is a eukaryotic system. So, taking a prokaryotic system may not be wise, therefore, one has to choose for a eukaryotic system. Even then, if one choose yeast, yeast is a eukaryotic system, but even then yeast is a single cell eukaryotic system, and the yeast metabolism is entirely different from that what is happening in the plant. Although there are some positive sides with yeast, because they are easily amenable for genetic manipulation, they can be easily cultured in a shorter period of time. Okay, genome sequence information is available, and also yeasts they produces, there are in the past there are evidences that they produced several proteins in stable amounts. Not only that, they efficiently undergoes they efficiently take the job of doing post translational modification of the protein, so that the enzyme protein will be active for the catalyzing the biochemical reaction involving the formation of natural products. So, it is known to be a good platform for metabolite production. So, that is the positive point, but the important point to consider that a plant pathway involves so many organelles. Okay, in yeast organelles are there, but it is not like the plant cell. Plant cell is much more complex. So, whether yeast will be capable of producing the metabolites or not. What I have said at the beginning of the class, when we talk about the metabolic engineering strategies, there, yes, it is possible that you can put a single gene in yeast and grow the yeast cell and then feed with the substrate, it produce the product, or you can take only a few genes, make a what you call the operon, and then operon is maybe a prokaryotic word. You can make a gene construct consisting of a few genes, and then if all these 4-5 genes function, and then even then the enzymes will be produced, but it may not get the substrate, because yeast are not meant for producing this plant metabolites. So, in that case you always have to feed the substrate to get the product. So, that again, if the substrate is expensive, then this is not feasible, this will not go to the industrial scale. Okay. So, what is showing in this side, that you should know the pathway, identify the candidate genes, identify the biosynthetic pathway, and then develop an engineered strain. Pathway may be reconfigured, then development of expression modules, fine tuning of the medium culture environment, and metabolic flux optimization. So, all these things are required, and if one is successful in doing all these things, then eventually the process can go to the industry. So, it can be grown in the bioreactor for harnessing the product. So, these are the important points to consider. So, now if we think of engineering yeast for improved production of putrescine. So, what we need to know first of all, that what is the native putrescine pathway operating in the plants and operating in the yeast. So, the yeast pathway is bit different than that of the plants, and there are so many genes involved ultimately for the production of putrescine. Okay. And if you want to express the plant gene, or if you want to transfer the plant genes for putrescine synthesis in yeast, then one can take these genes ADC, then this one, and along with that there may be required another microbial genes, so that the pathway may operate inside the yeast. So, in other words, say for the sake of argument, yes, it is possible to produce putrescine by heterologous expression of plant and bacterial combined pathway, and putrescine is produced. But putrescine is only the early step of the tropane alkaloid. So, that is not, that is a long way to go. So, next point is that engineering a microbial biosynthetic platform strain for de novo production of alkaloids. So, these I have to explain now, what is the meaning of de novo. So far what I said, that what I have discussed in the metabolic engineering strategies, that you can produce a short pathway, you can express a short pathway in yeast or in microorganism, and then the pathway will express it, produce the active proteins, but it will not get the substrate. So, substrate you have to do the feeding, and what I have just said, that if the substrate is expensive, then that will be of no use. Now, here de novo means you are not giving any specific substrate required to run that biosynthetic pathway. What you are giving, basically you are just growing the yeast in a medium containing glucose. That means, you are only supplying the primary metabolic product or glucose, because for growth of yeast you need to add some carbon source, and the yeast will utilize that for its growth, and it will drive the pathway towards the tropane alkaloid. So, this is a great challenge, and the beauty is that if one is successful, then you need not have to bother about the expensive substrate. So, a group in Stanford, so they actually decided to break the shell, and so this group was led by Professor Smolke, and Srinivasan was a PhD student from Canada, moved to Stanford for doing PhD funded by Canadian government, and they actually did this work. So, this I came to know after reading the paper. Okay. So, so engineering a platform strain for putrescine overproduction. So, the first one is that module 1, where arginine and polyamine metabolism genes are produced, genes are transferred, and as a result of that putrescine is produced. So, this is the module 1. So, the module 1 one has to attempt, and if it is fine and then module 1 is successful, then the engineered strain will produce putrescine. So, next to the see the module 2. Module 2 is basically to make tropane. So, how they have done? So, they have basically hired several genes from the plant sources, and so and these genes were subsequently transferred and expressed in the yeast system, and as a result of this, if as a result of their success, they could see that the tropinone and tropane can be produced in the yeast. So, in this case optimization of de novo tropine biosynthesis yeast is important. So, here in earlier view was that hygrine is produced and from hygrine tropine is formed, but the new view said that hygrine may not directly involve in the tropine. So, what is important that N methyl pyrolinum by the action of a polyketide synthase followed by a cytochrome P50 and a tropinone reductase, ultimately it makes tropine from N methyl pyrolinum. So, this pathway needs to be engineered in the yeast, and module 3 is basically phenyl propanoid pathway. So, this is something new they wanted to see, because one important point is that the phenylalanine is required to make phenyl lactic acid, and that eventually makes the joins with tropine and makes this hyoscyamin. So, phenylalanine in order to modulate that pathway, PAL gene and 4CL gene was also transferred in the yeast. So, and that way the module 3 was successful, and module 4 what they have done. So, they sometimes they once they made an ambitious attempt whether they can make cocaine in the yeast. So, cocaine is basically produced in the erythroxylon. So, there they have taken a erythroxylon cocaine synthase gene. So, which and they put it in the yeast, and what it does actually it joins tropine and cinnamoyl CoA, and as a result of this enzyme catalyzed reaction a new product is formed which is called cinnamoyl tropine. So, this is basically not known to produce in the plant or microbial system till date. So, but this has been produced in the produced by the engineered yeast. That means, now they joined this module 1, module 2, module 3, module 4, and as a result of this joining what we see, this whole pathway. So, where starting with glutamic acid and then it makes putrescine, then from there it makes tropinone and tropane, and this tropine joins with cinnamoyl CoA and it makes cinnamoyl tropine. So, engineered pathway for de novo biosynthesis of tropane alkaloids. So, now tropine itself is a it is basically the tropane alkaloid. So, here in this publication they successfully declared that tropine can be produced in the yeast by transferring the genes from yeast source, maybe one from microbial source, several from plant sources, altogether around 15 genes were transferred, and few yeast genes were suppressed or made deactivated. So, that yeast other metabolism can be stopped, and as a result of that yeast will follow this pathway of metabolism, and as a result what we see that yeast produces tropine, along with that it also produces a cinnamoyl tropine. This is a novel product. So, that is indeed a breakthrough happened, but they were not happy with this. Their ultimate aim to produce the hyoscyamin and scopolamine. So, now the point is this that the eukaryotic system is complex. So, and as I said, the pathway, when part of the pathway is operating in the cytosol, then maybe it moves into the Golgi bodies, then maybe it before moving to Golgi bodies it may go into the endoplasmic reticulum. So, endoplasmic reticulum membrane may contain some enzyme which helps to convert, and then it goes to the Golgi body, then it goes to the vacuole, then vacuole in the vacuole some conversion occurs. So, in order to go inside the vacuole there need to have some the transporter proteins. So, these all to be active. So, that means several major steps need to be cracked, including maturation in the endoplasmic reticulum lumen, in the Golgi body, trafficking to the vacuole membrane, vacuolar substrate import, vacuolar substrate import, and then finally, the product export and cellular tropane alkaloid export. So, ultimately the tropane alkaloid produced should come out from the cell and it should accumulate in the medium, then only the process will be economic. So, this all needs to be achieved. So, and they have all achieved this by formulating 5 modules. So, module 1 and module 2 was achieved before, and that was published in the Nature Communications. So, in 2019. So, here I just briefly tell, in module 1 glucose is converted to putrescine, by metabolic steps in the mitochondria and cytosol. So, this is mitochondria and here is the cytosol. In the module 2, the enzymatic reactions in the peroxisome, here is the peroxisome, and the membrane of the endoplasmic reticulum. So, that plays role in catalyzing the conversion of putrescine to the tropine. So, then the tropine comes to the cytosol. In module 3, which occurs in parallel with module 1, is basically glucose and amino acid phenylalanine are converted into phenyl lactate acid glucoside, because that is required eventually, that joining with the litorine and makes the hyoscyamin. So, that is basically taken care in the module 3, which is operating in the cytosol, and module 5 this says that tropine and the PLA glucoside are transported into the vacuole together and converted into litorine. As I said, the PLA glycoside and tropine converted into litorine, and then litorine is finally subsequently converted into hyoscyamin and scopolamine, that is by module 5. And finally, the module 4 is basically which occurs in the endoplasmic reticulum membrane, litorine is converted into hyoscyamin and then subsequently to scopolamine. This is the way they describe module 1, module 2, module 3, module 5 and then module 4. So, that is the way I have discussed this, and this paper published in the Nature. So, this is I have taken from a News and Views article published in the same issue. When an outstanding paper published in a particular issue, the editor asked another expert from a different place to read the paper and make it as a simplified version for the non-specialist like us. So, it was much easier for us to read this one written by Montano Lopez and Jose Avos to understand what is happening, and what I understood I just explained to you, and the original paper basically published in the same issue, based on that only this article is written. So, this is the one, this is the original drawing which is given in the Nature. So, this is a much complex one, here all the modules are described. So, this is basically showing the modular pathway construction for scopolamine biosynthesis in yeast. So, indeed they were successful in producing scopolamine, but the level of scopolamine or a hyoscyamin what you find in the plant, the level is less. So, that means that more optimization is required, but otherwise this is the very successful achievement that indeed de novo tropane alkaloid synthesis can be made into the yeast simply in the engineered yeast by putting around 20 to 25 genes inside and simply feeding the yeast with a medium containing glucose, and the normal metabolic pathway operates. From there it goes to the tropane alkaloid pathway and then it forms the desired alkaloids. So, this is the end slide of my lecture, and with this I will end the tropane alkaloid. Thank you very much.